This is episode 16 of the Immunology Podcast, T-Cell Regulation with Dario Vignali. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Dario Vignali from the University of Pittsburgh on the podcast to talk about his work studying T-cell regulation and function in cancer and autoimmunity. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to Immune Regulation News, a free weekly newsletter brought to you by the STEMCELL Science News Program. Covering research from the regulation, suppression, and modulation of the immune system, Immune Regulation News keeps readers current with the latest news, research, policy, events, and jobs relevant to the immunology community. Subscribe for free at www.immuneregulationnews.com.
So Brenda, you sound a little different today.
Oh, you noticed? I have some common cold on me today.
Okay. So you didn't decide to take up smoking aggressively in the last week or something.
No, I'm not that desperate yet. So it's just some good old rhinoviruses.
Yeah.
Yeah.
Well,
Yeah.
now that everyone's been wearing a mask for a year, we're developing immunological debt. And so they're seeing, especially with kids, that people who have been sequestered away haven't had their usual exposure to the milieu of human existence. And as a result, people are getting more sick now because we've all been hiding away from all those antigens for the last year.
I agree. It was gone, like everything was gone for a year and something, and then he came back with a vengeance. Everyone's sick. Everybody is terrible.
It's almost like we're designed to exist in this antigenic environment and hang out all the time.
Almost, right?
Who knew? Who knew the immune system did so much work on a daily basis? So underappreciated.
Seriously, we don't give it the credit it's due sometimes, but we try in this podcast. We try.
Which we try, yes. Well, do you want to start with a paper about something that we need to appreciate in the immune system?
Yeah, okay. I want to start today talking about how it's important to appreciate the immune system and how often we have ideas that sound great in paper, but they don't really work out that well.
Well, as a postdoc, you should know that very well, right?
Yes, of course. But it's nice to see other people having these issues too. So I just want to share with you a paper today that talks about the results of a phase 1 trial for patients with B-cell acute lymphoblastic leukemia using dual targeting CAR T cells. The paper comes from the lab of Martin Pulley and Persis Amrolia from the University College London. First authors Sean Cordova, Shimobi Onuha, Simon Thomas, and it's titled CAR T cells with dual targeting of CD19 and CD22 in pediatric and young adult patients with relapsed or refractory B-cell acute lymphoblastic leukemia, a phase 1 trial. And it's in Nature Medicine. So we all know that BALL can be a very devastating disease, and often patients can be successfully treated with chemotherapy, but many times they relapse. And nowadays, for some years, a novel treatment option for these patients has been CAR T cell therapy. We have 2 or maybe 3 already approved therapies using CAR T cells that are targeting a marker on the surface of these cancer cells because they are derived from B cells that express CD19 as the same as normal B cells. Also, CD22 has also been used as a target for CAR T cell therapy. So these receptors are picking up this molecule on the surface of the cell. The problem is that often patients relapse after treatment with CAR T cells. Usually the initial response is many times really, really good, but then eventually there is an escape, some clone escapes by downregulating the expression or eliminating the expression of CD19. And in the case of CD22, it's pretty common to see a reduced antigen density to the point that the CAR T cells cannot induce cell death anymore. So what they thought, the logic behind this trial was to say, well, if we are then targeting both CD19 and CD22, then it's less likely that you'll find clones that are downregulating both things at the same time to escape the T cells. And so that's what they do. They generate a T cell product that has two CARs that are expressed on the same cell from a bicistronic retroviral vector, and that they have costimulatory domains, which are not common for this the current generation of CAR T cells that we use now. They have a costimulatory molecule that improves the efficiency of this CAR molecule, and they're using OX40 and CD4-1BB. 4-1BB is used in one of the already commercially available CAR T cell products, and OX40 is used as another receptor that's also signals to the TNF receptor family of proteins. And so they actually did this on many patients. They recruited 23 patients. So it's very interesting to see the kind of numbers that these phase 1 trials can get. They recruited 23 patients, but they could only generate 19 cell products from 20 leukapheresis that they obtained from the patients. And actually, in the end, 15 patients were infused with the cell product that they call Auto3. And they use some interesting automatic platforms for generating this product, which I think is more and more is being used for improving and accelerating the production of these individualized cell products. And what they see, they have a dose escalation phase in which they put different amounts of cells and reaching a similar dose to the traditional CD19 CAR therapies. And although they don't see, so what they see from these patients is one of the main issues, or a really important issue with CAR T cell therapy, is the presence of CRS, so cytokine release syndrome, and many patients suffer quite gravely from this and need treatment with, for example, antibodies against IL-6 receptors and things like that reduce the action of these cells, becomes so active that they can actually kill the patient, the therapy kills the patient. One of the things they see is that in this, as they escalate the dose, and sometimes they even split the dose in two sittings, they prevent the appearance of severe CRS, which is always a good thing. But in the end, they don't have a much better overall response. So they have a really good initial response rate. Most patients have a reduction in the amount of tumor cells, but in the end, most of them relapse again, and they see that some of them don't express or express less of some of the markers, like CD22 or CD19. But in this case, the main issue they observe is that the cells just don't persist in the patient enough. And in the end, they have some patients that, for example, have even normal B cells present. So this shows that often there's so many different angles that you need to keep in mind for these clinical trials. So what the authors, they end up concluding is that the problem is that the cells are just not staying in the patient for long enough, which is a shame. So they have only one patient from the 15 that is still in remission, that it hasn't relapsed. Other patients have to start other treatments. Some patients died. Most patients had a response, but it was not very durable.
That's a bummer. But are they looking at ways to modify the cells so they live longer in the host?
Yeah, I think that's one of the holy grails of cell therapy in general, because that is the problem with many other CAR T cell products as well, and other deep types of T cell products in general. Oftentimes, they just don't persist in the patient enough to mediate their function. So there are many things that you can do. You can either try to select cells to start your cultures with that are known to be better at generating cells that are long-lived. So this concept of the stem cell-like T cells that have a potential of proliferating longer and grafting better. Also using some metabolic interventions in the culture that reduces the amount of terminally differentiated cells from these cultures. Some cytokines have been also, IL-15, IL-7 has been associated with better cells that are better capable of engrafting. But I have to say that the jury's still out on what is the best way of doing this.
Yep. Well, always more work to do.
Yeah.
All right. Well, switching gears a lot. I said the segue here is that this paper is the opposite. It's almost just it's a descriptive paper of things that we kind of knew, but they went in more depth on. And so I've been pondering this one a lot. It's called Parallelism of Intestinal Secretory IgA Shapes Functional Microbial Fitness by Tim Rolaneski is the first author. Last author is Andrew J. McPherson. And so it's in Nature. And I don't want to pick on the work of the paper. I think it's really important work, but I don't know why it made it to Nature in terms of it being 100% descriptive. So they were able to, look at IgA in the intestine. So dimeric IgA crosses mucous membranes in response to all types of non-pathogenic microbiota, causes most of the antibody production in mammals. And we know that's really important for a lot of things. They haven't done a lot of monoclonal hybridomas at this point. And so what they did in this paper is took recombinant dimeric monoclonal IgAs and mapped out how they functioned in response to microbial colonization with a single microorganism that required a nutrient that you have to supply exogenously to keep it growing. So that's how they can control the microorganism being present in germ-free mice. So it's a lot of work, right? Germ-free mice with the monoassociation and then inducing of these monoclonal IgAs and understanding how they work. They would look at how the mice reacted to that single organism and found that different mice created different epitopes. So it's kind of like we were talking about last week with that variability, that last time we chatted, with the variability that you'd see across responses. So you're seeing the same variability, right? You're seeing the same with a known antigen, different people and your own person makes different antibodies to that antigen, as we'd expect, different epitopes. And it's not the same from person to person or mouse to mouse in this case. But they say that even with the same antigen, you get different epitope specificities. But what they did do was interesting, is they showed that depending on the epitope specificity, you had different microbial metabolic alterations that would happen downstream of that. So if these epitopes will bind to different things, different antigens in the microbial milieu, and then alter the microbial metabolism. They also found that these antibodies would surface coat and generally reduce their motility, but also limited bile acid toxicity. So they made the bacteria move less, but also limited the ability for bile acids to damage them. And so they're kind of the end point of this is that the intestinal IgA system, for any given microbe, and they used one single one, has lots of different things that happens. So it affects on the microbe's ability to uptake different carbon sources. It affects their ability to be killed by bacteriophages. It affects their motility. It affects their membrane integrity, and different epitopes of IgA to the same bug, to the same root antigen, generate different responses. And so it's really a tour de force in mapping this and how they do that. So they take these antibody deficient mice and reconstitute them with the feces, and then they reconstitute them as well with injection of the antibody. So they basically take antibody deficient, they put in the IgA that they have a clone from another mouse, and they can do those studies. And they generated those clones by doing the original studies in the germ-free mice as well. And so it's a lot of tour de force work, really, again, germ-free mice, antibody deficient mice, putting those together, doing a lot of epitope mapping, figuring out how they all respond to each other. So maybe that's why it ended up in Nature, but it's also very descriptive. I don't think it gives me a lot of new aha moments. It gives me a lot of, okay, yeah, all the things we knew antibodies could do in the gut to different microbes are happening. I guess the one upshot is that even a single antigen, so the plethora of antibodies generated to a single antigen or single organism provides numerous parallel mechanisms depending on which antibody is generated, which epitope. And that gives us diversity. So the diversity in epitope generation that our antibody, our adaptive immune system has, generates a downstream diversity in response. And so I thought it was very interesting. I'm wondering how this ended up in Nature versus like Nature Immunology or Nature Communications, or kind of that one step down, highly descriptive tour de force work, but I'm not sitting there with like my mind blown open by a massive change in the field. But maybe I'm missing something.
I guess what it's interesting is that if all these mice are genetically identical, then you're really gonna support the idea that all this variability comes from the environment because they are genetically identical.
Well, and their recombination, right? So, we know that
Yeah.
antibody recombination is deliberately promiscuous and non-accurate. And so even the same, twinned mice will produce different antibodies to the same antigen because the hypervariable recombination is going to generate different results from organism to organism.
Yeah. But they are based on similar initial chains.
Yeah.
So like you have the same kind of canvas in which you place these mutations, but you would expect that there's going to be preferential groups of mutations because that's what the physics of the interaction of the antibody and the antigen
Right.
tells you. And the starting material is always the same. So because I'm thinking, for example, in the case of TCR receptors in human populations that are so genetically so variable and then have so many different MHC classes that will really limit the type of presentation of your peptide. And then you see, well, there's so much variability from the gene to the germline that, yeah, you're just going to end up with very different receptors and very different antigenic presentations. But I guess, of course, B cells and antibodies are detecting antigens that are not being presented. But I guess, are there polymorphisms or things that could be modified between people that have already different starting material before the hypermutation?
Yeah, I mean, I think that's part of it. And like in this case, it's the same one, and they do see that it drifts, right? So they see that you have the original change before the hypermutation, and then they see as well, though, that it's they see a cluster, right? It's not like everything's just a complete schmear. There's definitive clustering because you do have the same inputs, genetic and antigen wise.
Right.
And I think at one point they restricted it and did it before the hypermutation stage as well as part of their model. I have to go back and dig here. If I remember right, they also did that before hypermutation and saw no changes in it.
Before the hypermutation, you're going to have clusters of selected combined segments that are more likely to be specific against this microbial antigens.
Right. Well, they looked at it and found that after introducing the antigen, this very specific E. coli that doesn't grow without the, extra supplement, that there's no increase in somatic hypermutation to it. It doesn't drive.
Okay.
There's no further. So you don't have the secondary hypersomatic mutation step in this case. You just have the regular like recombination.
Yes. Okay. And that's because the mice are deficient, or just because it's so good, there's already such good binding that...
I think there's good binding in this case, and it's an IgA system.
Yeah, of course. Well,
I mean, they do some, but it's not the same type of renewal feedback, somatic hypermutation you get by hanging out in the lymph tissue all day.
All right. Well, I don't know. I haven't read the paper, so I'm not sure, but it seems like they did a lot of work, and I'm glad that they got recognized by a high impact paper. So more people will be bringing out that very interesting basic science to the big leagues. You're not convinced. That's okay. I will move on to my second paper of the day, which I was very happy to read. You might remember last year when the results from the phase 3 trial from the AstraZeneca vaccine came out.
Yeah.
That there were a lot of issues in general with AstraZeneca vaccine, and one of the latest they had was that when they showed the results from the phase 3 trial, they had to admit that they had made a mistake when giving the doses to a cohort of patients, because apparently they realized after the fact that these patients actually received a half dose as the first the prime vaccination. But the funny thing was that when they actually looked at the efficacy of the vaccine, this group with a lower dose was substantially better. So the numbers were quite impressive, because they had something like 90% of efficacy for this quote unquote mistaken low dose, whereas the standard dose that they actually carried through was around 62%. So like the counting all the vaccination was around 70%, and that's where this initial efficacy of this this vaccine came out. And the fact that AstraZeneca didn't seem to really pursue further these results really bugged me, because it makes so much sense that if you have a better efficacy with less amounts of vaccine, that has to be a win-win situation. And there's a lot of reasons why AstraZeneca did not continue with this, and I'm not going to start discussing them now. But what is really cool is that there is this paper that came out in Science Immunology in which they actually try to model the effect of using lower doses to prime the vaccine responses to higher doses, and what's the effect on the induced immunity in this case in mice, in murine patients. So this paper comes from the lab of Pablo Peñalosa McMaster at Northwestern University, and the first author is Sarah Sanchez, and it's titled Fractioning a COVID-19 85 vector vaccine improves virus-specific immunity. So they do this work on one particular adenoviral vector, which is not the same from the Oxford AstraZeneca vaccine. They used a chimpanzee-derived vector, adenovirus vector. And in this case, they're focusing on a different one, on a human adenovirus, which is adenovirus vector 5, which is known to be very highly immunogenic. And it's actually present, it's actually used in two different vaccines that maybe most people in the developed countries have not heard a lot about, which are the CanSino and the Sputnik V vaccine. In the case of CanSino, it's a one-dose vaccine similar to the Janssen or the Johnson & Johnson, and the Sputnik V uses a two-shot, a two-shot regimen. And the first one, they use the 80-25, which is the same as the Johnson & Johnson vaccine, but they use the 85 for the booster shot. So it is used as a vector for COVID-19 vaccines. So what they do is that they test in mice how they compare, basically giving the mice two standard doses, 4 weeks apart, or a first dose, which is 1,000-fold lower, and then a standard dose for the booster. And so they look into the responses and the CD8, the expansion of CD8 T cells specific against a viral epitope. They look into cytokine production and then the quality of the T cells. And after the first shot of what is as expected, they see that less vaccine results in less expansion of T cells specific against the virus. They see cells that are less active. But what they see, which is very interesting, is that after the booster shot, which is the same for both arms, the mice that had been initially vaccinated with a lower dose perform substantially better. They have more expansion of secondary expansion of CD8 T cells against the virus. They produce more interferon gamma. They produce more granzyme B, more Ki. They were also more Ki67 stain. These cells look pretty good. And what they see is that when they look into the cells after the first dose, a lower dose generates more cells with what is usually considered known as a central memory markers. They're markers of such as CD62L and CD127, which is the IL-7 receptor alpha, that are associated with cells that are long-lived and capable of kind of quiescently persisting in the organism. They look also that having a lower dose at the start generates more germ cells in the spleen, more B cells from the germinal centers in the spleen. They also have more higher titers of antibodies and of more a better neutralization titers for the serum of these mice. So in general, they are much better starting with a lower dose. They also look that by having a lower dose, there's a lower neutralization of the vector itself. They observe that a previous high dose of 85 generates that upon the booster, the cells that are picking up the new 85 vector are more likely to be attacked or to kind of disappear because they're expressing its epitopes from the 85 than when you start with a lower dose. So they don't see clearance of the virus itself, or they don't see a reduction in the transduction efficiency of this virus, but they see that the cells that are transduced survive less, and therefore they cannot present the antigen for long enough. They end up looking also for a different model. You also test a model in which they are expressing a different viral epitope, in this case, the GAG protein from simian immunodeficiency virus, and they see the same lower doses at the start result in a better response after the prime with the same amounts. So I thought that was really cool, and I really hope that works like this will start kind of nudging people to start using lower amounts of the vaccine, because I think that in general, the side effects of also the vaccine, and also in the case of the RNA vaccines, they're probably related to such high doses that were done because often people think that more is better. But in this case, it looks like less is more.
I was going to say more is better seems to be the standard modus operandi. So it's good to see that that's not always the case. But interesting that, I mean, you would think AstraZeneca would go after this, because then they can like make more doses per batch of production run.
Yeah, but in the end, they will be selling less vaccine. So if you want to be cynical,
no, you sell the same amount of vaccine. It's $10 a dose. You're just putting half the drug in it. And it's more efficacious.
Could they get away with it?
Yeah, absolutely. Absolutely.
But no, they're like $2 a dose. So they're selling at a cost to many.
Right. Like, so now they can drop how much you can sell it at a profit then by using less drug product in each. There you go. I mean, like bing, bang, boom.
Yeah. Yeah, I think it makes sense that also it would reduce side effects, and the problem is now, and that's the case of AstraZeneca, but also other vaccines such as the Johnson & Johnson that uses the 8026. You cannot really vaccinate twice, and like the other ones, because people usually think that because you already have pre-existing immunity against the vector itself, having this high dose will get you prime against the vector, and this is what they observe in this mice, that they also see some loss of efficiency because the vector itself is being targeted. And I think that maybe by having a lower initial injection, you can maybe be able to successfully use a booster without losing the efficiency, the efficacy.
Yeah, no, I think it makes sense. I'm surprised they haven't jumped on it, but who knows? Sometimes you just get stuck with what you're using.
Yeah, I mean, it works, don't touch it. I think that's what they're kind of, it works, now a little bit, no, but quote unquote, because with all the variants and such, I think it is time for a revamp, but we'll see if the incentives are there. I'm not sure.
All right. Well, time to hop on to our last one. This one is IL-33 induced metabolic reprogramming controls the differentiation of alternatively activated macrophages and the resolution of inflammation. It's a German group in the lab of Gerhard Kronke, and Maria Voss is the first author. I like this paper a lot. It has all the things I like in it: cytokine signaling, a specific immune population. I like macrophages and wound repair. And it does a deep dive. It does have one weird thing to it that I'll get at, but I think that was a reviewer shoving their opinion into a paper. So basically they show through this mapping out, we know that there's all these alternatively activated macrophages, or AAMs, that do both inflammation, resolution of inflammation, and tissue repair, but how they're differentiated and how they function was not well established. They show here through a series of great experiments that IL-33 is the primary inducer, and IL-33 does then two things. It does an NF-kappa B downrated response through MyD88, which leads to inflammation. And that inflammatory response plus the MyD88 signaling itself also regulates something called UCP2, which is an uncoupling protein for the mitochondria to cause proton link to lead to more efficient TCA cycle. And as a result of that process, you get more GATA3. And GATA3 transcription activation leads to anti-inflammatory behavior, resolution, and repair. And so if you block UCP, the entire effect goes away. They show it's a metabolic function. So you have a shift in metabolic state that occurs not early. So you have MyD88 and FKB activation. That's one step, right? So that, that's still a transcription factor. That's still 4 to 8 hours, right? But then after that, you have the secondary, not change in UCP transcription, but it'll decrease degradation. That decreased degradation leads to metabolic shifts in the macrophage. And that then leads to these other downstream effects, a high GATA state and all the other benefits. And so they do all the things. They do the right knockouts to show this. They show how they do the seahorse assays to show the metabolic shifts. They do single cell RNA-seq, although I'm going to slap them on the wrist because they did an N of 1 equals 1 mice each for the knockout and wild type in this case, which I was impressed that this paper, which is in Immunity, by the way, got away with N equals 1, comparing GATA3 knockout and wild type mouse for the single cell RNA-seq and the pseudotime. I don't know how they did that, but they did. So kudos to them for saving money on their grant budget. But it was well done, and they really mapped out everything along the way. I will say one thing that confused me in the paper, but I kind of started picking up on later, is they talk about IL-4 really early. It was like this alternative pathway. And my guess is that IL-4 can also induce these alternatively associated macrophages, but they just like shove it in the middle of talking about figure 1. I'm like, oh, we also looked at IL-4, and then they shove it in a few other spots. And I'm wondering if that was reviewer number 3 or reviewer number 2, or whichever reviewer you want to blame today. They really wanted them to talk about IL-4 and say just shove it in a few spots to appease the reviewer. That's my guess, because it doesn't seem to fit the rest of their narrative. And they could have just said, we're looking at IL-4 independent, but they like, show IL-4 does this, but not this and not that. And here's some RNA-seq comparing the difference between IL-4 stimulation and non-RNA IL-4 stimulation. So maybe with RNA-seq, they, hopefully the reviewers didn't make them do it was bulk, not single cell. So maybe not, but it's cheap enough they could have asked. I just don't get where it just popped up from. I'm not an expert in AAMs and IL-4 signaling in relation to them, but they basically show that it's separate. So that's, it's clear they specify that it's IL-33 and IL-4 induce separate phenotypes. So I don't know if that's something they had to put in to like early rule it out, or maybe they were looking at that and that was the original project. And then they realized that IL-33 had something interesting going on that was different than IL-4 and went down that. And so that's how they had the paper. And then they wanted to weave in the IL-4 data back into it. But again, really cool. IL-33 has this early and late stage. And what's neat about this late stage kinetic is it's driven again by these metabolic changes. So you have to have this protein accumulate. The protein accumulation leads to metabolic changes. The metabolic changes leads to GATA3 accumulation, which is another transcription factor. And so if you think transcription factor 1, NF-kappa B, is the first hit after signaling, you have all these other steps to get the second transcription factor. And that's how you build in an early and late stage kinetic into a signaling pathway. And that makes me happy to think about things. Anytime we can discuss cell signaling and kinetics, enzyme, thermodynamic, I don't care. I like kinetics. At any point in my life, I'm a happy person.
Good. I'm glad this paper makes you happy. But yeah, I think probably because IL-4 it is usually associated with this alternative activation of macrophage, I think, and that's why they have to put it there. I would not be surprised if they were not necessarily looking at IL-33 in the beginning, but it's really cool to see how this by looking at the metabolism of these macrophages, how they seem to really affect their differentiation. I think that has been already described often, so it is known that this alternatively activated macrophages have a specific type of metabolism.
Yeah.
But it's interesting. I don't think we knew that IL-33 could trigger this through this mechanism by affecting GATA3.
Yeah. Well, by affecting it affects the accumulation of a protein, and that protein accumulation leads to GATA3.
Yeah.
So the metabolism causes the GATA3, and that was interesting to me. Like, it was neat to see. Well, Brenda, we're going to be speaking to Dr. Dario Vignali here in just a few moments. But before we get to that, Ensure reliable results with your immunology research. From primary human cells to cell isolation kits, cell culture media, supplements, and antibodies, STEMCELL Technologies provides the tools you need for every step of your immunology research. Interested in cell isolation? Use EasySep to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Cells are viable, functional, and immediately ready for your downstream applications. Learn more at easysep.com.
Joining us today is Dr. Dario Vignali. He is a distinguished professor of immunology at the University of Pittsburgh School of Medicine. And Dr. Vignali has made great contributions in the study of T-cell regulation and function, and particularly of Tregs inhibitory molecules and T-cell signaling in the context of cancer and autoimmune and inflammatory diseases. Dr. Vignali, thank you so much for joining us today.
Thank you. It's my pleasure.
Thank you for being here. So I'll just jump in real quick. Here at the Immunology Podcast, we discuss which immune cells are our favorite. Brenda's very clearly on it's a T cell. My answer is it's an enterocyte, but that's for other reasons. T cells are pretty close second for me. So I wanted to dive in right away and, give us the top thing about Tregs, because, we Tregs are the T cell, the control, the bad behavior, as I like to think about it, of other T cells. And it's been a field that's been appreciated and evolved, and we've learned about oral tolerance and their importance in autoimmunity. And yet, the more I feel like we learn about Tregs, the less we know about Tregs.
Yeah, it's a great way to put it. I mean, I think I'm, a little bit of a T cell chauvinist in that I'm really interested in T cells in general, not lacking in appreciation for the importance of all cells in the immune system, but spent most of our time focusing on T cells, and as a subset, regulatory T cells. I think the things that excite me the most about these cells is the sort of controlling capacity they have, the sort of, they're often considered the conductor of the immunological orchestra, and really can guide, not just from a suppression perspective, but from guiding the type of immune responses you can get. I think there's a number of studies that have shown that.
A lot of your research on this field has been on understanding the regulation of T cells and also the regulation of regulatory T cells. I was very interested in talking about, for example, this molecule LAG3 that you have worked a lot on. Yeah. And other kinds of molecules that are related to Tregs, for example, neuropilin-1. Also, you found the correlations between the expression of this marker and the function of Tregs. So maybe you can talk a little bit about what regulates Tregs and how does that correlate with other T cell subsets and conventional T cells?
Well, that's a great question, but obviously not one that can be given in a short, simple answer. I think like most cell types, regulation is complex, and we need it to be complex because if it isn't, then it's too easy to subvert. So, regulatory T cells receive cues from the microenvironment, which shapes their behavior. That can be cytokine driven, that can be cell surface molecule driven. Some of those are shared amongst others. You mentioned LAG3. So a lot of the inhibitory receptors can shape Tregs in the same way as other cells, but they also have some things that are unique, like neuropilin. So I think it's really multifactorial. And the challenge is really trying to define the ones that make the biggest impact on them, because those are obviously the ones that are of most interest from a translational perspective.
Do you have a Mount Rushmore of like the top 4 or 5, as you said, that really keeps standing out as a theme?
Yeah, I mean, FOXP3 is, from a transcriptional perspective, number one. There are obviously others that people are interested in, and I think there's still a little bit of an iceberg there in terms of things that haven't been interrogated to the same extent. Beautiful papers came out recently regarding TCF1 from Gunari's lab and Kazai. And I think there's probably other transcription factors that are important. From a cell surface molecule perspective, obviously we're big fans, biasly, admittedly, of neuropilin as being a key controller. I think that would be one of the figureheads in Mount Rushmore. maybe, and I think there's also other molecules that we just don't know. I think that the iceberg is a good analogy. As you pointed earlier, I think the more we understand, the more we realize we don't know. And I think that's still the case with Tregs. And then soluble factors. I mean, we know the cytokines will influence them. So that would be the third head. And the fourth, I think, is only just coming to realization, and that's metabolic control. Beautiful paper from Greg Delgoff that came out recently in Nature. I can brag about him because he was a former postdoc of mine. Did some of the original neuropilin work and showed that lactate is used as a dominant food source in tumors, which is really interesting considering the fact that glucose is considered, the primary fuel for highly glycolytic T cells. It seems that Tregs can use lipids and other molecules as food sources, which creates distinction between Tregs and effector cells. So those are your kind of four heads, so to speak.
Just for the members of the audience that might not be very familiar, what do we know about, for example, neuropilin or LAG3? How are they affecting T cell function, and why, for example, LAG3 is kind of a checkpoint inhibitor? What it differentiates this inhibitor or a checkpoint molecule from PD-1, for example?
Yeah. There are definitely similarities and differences, as is often the case. So LAG3, to start there, is an inhibitory receptor in the same way that PD-1 is. It controls TCR signaling, but it does so in a different way. And even part of that we don't know. We have a lot of unpublished data regarding how LAG3 works that isn't ready for prime time, but it's very, very different to PD-1. And that difference, taking a sidestep away from Tregs, is actually important for combinatorial immunotherapy. We want to combine things that function differently. And that's why, some of the more recent data, which we can talk about if you want, from a clinical perspective, combining PD-1 LAG3 inhibitors has been so exciting. But going back to Tregs, fundamentally, these inhibitory receptors are brakes, right, to use that analogy. But they're brakes in different ways. Some of as the foot brake, some as the hand brake. And so LAG3 and PD-1 don't really function in a way that's different in Tregs versus effectors. They slow them down. So if you remove them, you can release them. But all cells are prone to apoptosis. So if they get too energetic, they can die. So it's a subtle balance. Neuropilin's a little different. It seems to, at least in Tregs, control some of the activity through AKT and mTOR. And some cells love AKT. Going back to the metabolism angle I mentioned, T cells love AKT, whereas Tregs, they need some, shown quite nicely by Hongbo Chi a while ago, but they don't like too much, because then that actually limits FOXP3 activity. So these molecules kind of have similarities, but also distinctions in terms of how they control regulatory T cell and T cell function in general.
That's really interesting. I wanted to home in on that with AKT, and I know you've worked on SREBP1, so the sterol receptor and element binding protein for, which for people who don't know is a transcription factor that senses metabolism. And so I was wondering if you could dive in a little bit of that. I've done some work on immunometabolism in the past, and actually looking at the pathways that regulate phosphoinositide signaling in AKT, and realizing that they really are important tuners for all types of immune function. So I'm wondering maybe if you could dive in a little bit about that before we switch to some of your hints at clinical trials in terms of how metabolism, because immunometabolism is this budding field, what you've seen in terms of this, especially that Treg conventional T cell balance.
Yeah, I think it's a great question. The link and focus between cancer and metabolism, of course, goes back decades when, the development of the antifolates as the first anti-cancer drugs. And a lot of the focus has been on the tumor metabolism. And I think it's only recently, through work from Greg Delgoff and others, Erica Pierce has been very active in this area, Sukek, etc. Really trying to gain an appreciation that the metabolic processes of immune cells are also important. and how those are influenced by the tumor microenvironment, because, they're using glucose and oxygen, and so they are perturbed in the tumor microenvironment. But that also presents an opportunity, because we need to do two things. Firstly, we do need to energize the cells. It doesn't matter how many switches and buttons we turn on with regard from immune modulators. If they don't have the fuel and the energy to commit to their anti-tumor program, they're not going to function. So I think there's a lot of therapeutic opportunities there, but also things that are different in the tumor compared to other inflammatory sites are important as it relates to immune adverse events. Because, if we soup up immune cells in an inflammatory site, thinking about tumors as being inflammatory sites, we don't want to, for instance, induce IBD or other inflammatory things. So if there's something that's unique in the tumor that's different from a sort of a conventional, regular inflammatory site in other sites of the body, perhaps leveraging the fact that there's differences in amount of oxygen, amount of glucose, might create things that are a little bit more selective for the tumor microenvironment.
And on that note, what do you think are the most exciting current kind of translational trials or translational research really looking into moving this knowledge about so metabolism, regulation into the clinics?
Yeah, I think there's a sort of a plus and minus there. The plus is that I think there's really exciting opportunities, but a lot of those have not yet got into the clinic. And in fact, I think we're still grappling with, okay, now we've appreciated the concept. Now we have to think about developing targeted therapeutics. And that lag is sort of normal in the process of thinking about therapeutic developments, and maybe 4 to 5 years away, maybe less, maybe more. I think the only things that are entering the clinic is not so much things that might specifically target a metabolic pathway in a cell, but repurposing of drugs that can change the microenvironment. So, for instance, again, not keeping going back to my previous trainees, but Greg Delgosh showed that use of a repurposed type 2 diabetes drug, metformin, can increase oxygenation in the tumor microenvironment and promote an anti-tumor immune response. So those are things that can be put into the clinic, and there are ongoing clinical trials to try those combinations with anti-PD-1. So those are the things we can do quickly. Things that are going to be more sophisticated in the surgical are probably going to take a few years to get through to the clinic.
So I'm going to take us a step from clinic back to mice, because to be able to do the clinic works, you need good mouse models to figure out what's going to work and what's not. And this kind of goes to your more recent paper, which is frankly a really cool paper. So I just want to say that this mouse that you have in immunity is pretty interesting.
Yeah.
And people should read the paper. But I was wondering if you could give kind of a high level view of this, because you did this really unique combination of alleles to be able to finally target conventional CD4 T cells. And maybe you can explain, like in a sentence or two, the problem behind why you needed to do it, and then explain it. And then in the midst of this, if you could also let the audience know, like having bred mice myself, that there's a lot of work that's just like one sentence, like we made a mouse, which is, 3 or 4 or 5 years. So if you could even describe a little bit, kind of, so people can see behind the curtain, like what it took to, if you're willing to, what it took to get the damn alleles ready to go to then cross with each other.
Yeah, so you've touched on a couple of interesting points there. So first of all, I think the audience will appreciate that as we think about the scientific challenges, we really want to be focused on the scientific question and the hypothesis, right? Rather than, hey, this is a cool tool, now what are we going to do with it? But at the same time, I think we all appreciate that discovery is enabled by having the right tools. And I think one of the challenges is as we went through an era of generating a whole set of cool CRE tools, we started to realize that there are some subsets, whether they be T cells or others, that don't have a unique gene that can define just that subset. CD4 and FOXP3 were great, but how do we get beyond that? And there are no genes that are only expressed in CD4 effector cells. And so we have to start thinking about more sophisticated ways to create Cree lines that are restricted in their expression. And so we took a leaf out of a couple of studies which have used a process called allele conditioning to generate this mouse. So that in principle, it's just simply where you have one gene or one allele that influences the behavior of a second allele. And so we did this by using two different recombinases. First, we put in a flipase, which is functions in the same way as Cree recombinase, but just recognizes different flanking sequences, and put flipase into FOXP3. And then secondly, created a mouse that expressed Cree just in CD4 cells, but had that flanked by Fritz sites. So in the presence of FOXP3, that was deleted. So it was now not expressed in Tregs and was only expressed in CD4 effector cells. So that way we could have a Cree that's only expressed in CD4 effector cells. It does complicate things because now you've got two alleles that you have to breed in combination. And anybody who's done these sorts of things knows that if you then combine it with one or two floxed alleles, now you have a quad. And if you want a Rosa reporter, that's 5. And it's as you point out, it's easy to say we made this mouse, but to breed that actually probably takes about a year and a half. And we can accelerate all kinds of things now. It's amazing what we can do scientifically, but unfortunately, we've not found a way to accelerate mouse breeding. So, it used to take a year to make a mouse using recombination. That's how we started. Now with CRISPR, you can actually make them in 2 or 3 months, but it's still a long process.
Well, and I'll just point out real quick, CRISPR only sometimes works to get the Cree in or what have you, or the Flox tags, not the Cree, but the Flox tags. I've had real hit or miss depending on the gene.
So you're right. It's not as simple, but I have to say the technologies have improved a lot, and our hit rate is actually quite high. So the last couple of mouse strains that we've made, it's kind of secret because we just made it, a new Cree RT2 lock-in. We got 12 mice, and 3 of them were knock-ins, and they're correct. That was just with the first injection. So I think at least at our core, we're on a bit of a roll. Hopefully I'm not jinxing the whole thing and everything will fail from here on in. But I think there's been a lot of intense activity around improving Cree approaches because people know how powerful the technology can be, that it's improved substantially, even for putting in big things like Cree RT2 and GFP and things like that.
So you should ask him for a protocol, Jason, for future reference.
Or the core at a place I no longer work at, but yeah.
Well, maybe you could do a podcast with Sebastian Gringas, who runs Hours. He's a genius with these things, and he could probably tell you all kinds of cool tricks and tools.
There you go.
So kind of what are the kind of questions or what kind of approaches can you do with this mouse that you couldn't do with like the traditional CD4 log Cree versus FOXP3 Cree and see how those compare? What are your plans with this mouse?
Yeah, great question. So, I think one of the challenges of trying to manipulate things in CD4 is first and foremost the fact that you're also influencing Tregs, and they're often working against each other, so it makes the interpretation complicated. But I think for us, at least as it relates to the field of cancer immunology, the field has been totally focused, understandably, on CD8s. They're the primary drivers of anti-tumor immunity, and so it's understandable that we've been focused on that. But I think there've been a number of studies in the last 5 or 6 years ago, maybe people reopened Janeway and Reut and realized that CD4s exist. It's amazing how immunologists like to reinvent what they knew for decades, right? And CD4s are important. They don't only guide and educate B cells, but they also help CD8s, and they play a role in anti-tumor immunity. And we've not really interrogated them in terms of how they do it. Is it exactly the same as CD8s? Do they do things in different ways? And so to be able to address those questions, we need tools where we're surgically manipulating just CD4s to ask some of those questions.
I have one last question I really wanted to kind of bring up, and I think you're a great person to talk to this. I want to hear more about what is Treg fragility and how does that affect our understanding of Tregs, for example, in cancer or in other diseases?
Yeah, great question. Thanks for that. Of course, I could give a whole podcast on that, but to keep it short, maybe a quick comment about terminology here, just so the audience is, on the same page. So the term that's being used most frequently as it relates to regulatory T cells, as you know, is stability. So when people talk about Treg stability, which is an important topic, they're mostly talking about loss of FOXP3, that when you lose that key transcription factor, the Tregs lose a lot of their identity. And that can be a good thing if you want to subvert them in cancer. It can be a bad thing in autoimmunity. And indeed, lack of Treg stability is a concern. And it's also of translational relevance, particularly with people who are trying to develop therapies for autoimmunity. They don't want their Tregs to lose that identity. Not only because of the danger that, well, you don't have Tregs and they don't work anymore, but the concern that they may become something pathogenic because there is a greater sense of recognition of self by regulatory T cells, at least ones that haven't been engineered to have a specificity for some particular antigen. And so the concern that they might start attacking the body. So that's the whole stability thing. When we started working with neuropilin, we kind of originally thought, oh, they lose neuropilin and become unstable. But we realized that actually that's not the case. These cells maintain FOXP3 function. They maintain most of their Treg identity. So that we needed a different term, and so coined fragility, because they lose their suppressive capacity, but they maintain their identity. They maintain FOXP3 in that. So I think fragility from our perspective is an exciting concept, because if we can turn off the suppressive capacity of regulatory T cells without risking them becoming autoreactive to self, that might be the kind of holy grail. And so we're very much focused not only as it relates to neuropilin, but trying to identify if there are other things that will drive fragility but not instability.
For example, does interferon gamma, so you have both can induce fragility, but Tregs can also produce interferon gamma. How does that work?
Yeah. Thanks for raising that, because one of the more recent papers we had from a super talented grad student in my lab, who happens to be married to Greg, Abby Overaker-Delgoff, showed that gamma interferon is a key driver of Treg fragility. And neuropilin is trying to oppose that by giving them, more stability, I guess is one way to put it. But indeed, you kind of raise a bit of a conundrum, because Tregs do make gamma, not a large percentage, but they do. And we're still trying to understand what that means in terms of Treg function. Is it a sort of a auto-regulatory loop? Not an unusual concept in immunology, of course. Sometimes the cells will make the things that can turn them off for control. Or is there some other purpose for this? The short answer is we don't know, but I think it's going to be interesting to investigate over the next year or so.
So talking about the stability, which is a newer concept to me, I wonder if it answers a question of something that's always bothered me, which is there's no T cell leukemias, or there's no Treg leukemias, right? There's no, you don't have, I mean, sure, there's a rare case probably, but when we think about, lymphocytic leukemias of some form, we don't have Tregs. Do you think that could be related to the fact that they're unstable? So as soon as something goes screwy, without getting too deep in, that instability makes them flip out, and so you're not running around with clonal expansions of bizarre FOXP3 positive Tregs. It just doesn't do that.
It's an interesting question. Not something I have any hard data on, but it's interesting to hypothesize, right? There's a couple of things. It could be a numbers game. There's far less Tregs, so the frequency might be a lot less. I think the point you raised may be the most critical one, that FOXP3 is a repressor, and therefore for a leukemia to develop, it would downregulate anything that it sees as a repressor. So it may well be that if you, for instance, if you had a constitutive expression of FOXP3, that would simply prevent leukemic development or leukemogenesis, and therefore, tumor cells that came out of that would find a way to repress or delete or inactivate FOXP3 in some way, right?
Yeah, no, I think that's where my brain was going. Is I'm wondering if this concept's linking to like the clinical absence of something is often really important when you think about physiology. Like you just don't get some things, and so that tells you a lot about the biology. Yeah, thank you. I think Brenda, you're up to bat here.
Yeah, no, I just could talk about Tregs all day. I think the listeners have heard that many times from me. It is fascinating also to look further into how Tregs actually are doing the work and what are the kind of signals that they are actually giving. I think your research has really worked a lot on that. So we mentioned also things such as IL-35 that you also have been working a lot on that. And I think I really like the concept of understanding also the stability of Tregs and the kind of signals that give them identity and give them function. So it was very interesting to have this conversation. I thought it was really great. So at the end of our conversation, we usually like to ask a little bit of questions out of the box to our guests. And so what we call the fun questions of the end. And so we were giving you free range to ask. So we're going to ask, for example, if you were not a scientist, what would you be?
Well, it depends probably at what age you ask me that. Because when I was young, I wanted to be a football player, professional football player. That's soccer for the American audience. However, when I got to 5, I realized that wasn't going to happen. No, a little older than that, but no, actually, when I was young, I wanted to be a doctor, a physician. But sadly, I was sort of young and naive and didn't work hard enough at college, and so ended up taking the research route. But I think actually, in the end, I'm quite happy with the position I'm in, because I think one of the important things for research scientists to know and understand, and I'm sure many of them do, is that by developing therapies, we can impact the lives of far more people, right, than any physician could see on a kind of day-to-day basis. Not to limit the amazing things that our frontline workers do, but I think researchers can play an important role in that regard by developing therapies, diagnostic tools, ways to identify patients that respond to therapies. I think there's a lot of laudable things that we can do, and we should focus on that. And in this day and age of COVID, it's been absolutely remarkable to see how rapidly vaccines have developed, how amazingly potent they are. And it's a sort of a testament to immunology as a field and what we can bring to the clinic through therapies.
I very much emphasize with that point about what researchers can do, having been unable to decide, done both, and then in the end landed on the science side.
To add one more thing to what you said, Brenda, more recently, everybody has their kind of COVID-induced stories. Like, okay, you had the shutdown and you couldn't do anything else but do things remotely. So I actually started brewing beer. Going back to, college, you'd brew beer because, well, it was expensive and it was cheaper to brew it yourself, right? But now I just brew it for fun. So, what started off as a small 1-gallon thing has grown into, I'm doing, 5-gallon brews, putting them in kegs. I've got some taps in my bar. It's fun.
Can I order online?
Yeah, sure. But it's an extension of chemistry, isn't it? Right? As scientists, we like to concoct things. I think that's why some scientists like to cook. It's an extension of that.
It's microbiology.
Exactly. Yes, all the yeast, and all the different yeast esters that can be generated, and how that can create really fruity, hazy IPAs if fermented at a high temperature, and all that kind of good stuff. Very nerdy, but fun at the same time.
Absolutely. All right.
I want a beer. It's a very popular hobby among scientists, I have to say. I've heard a lot of, and I've tried a bunch of homemade beers, and they're pretty good. And they also have a special flavor, of like his person made it. It's really nice. So, cheers to that.
There you go.
Cheers to that.
And I have one follow-up question, which is, if you could have any superpower, what would it be?
Oh, well, I think, I don't know, peace and tranquility and lack of anxiety. I think that especially at the moment, people are getting very anxious. I think people's mental health is going down. And I think there are a lot of challenges nowadays, especially for younger people. And so if I had the power of turning people from half glass empty to half glass full, then I think that would make a difference, right? We can't always change the challenges that we face, but we can change our perception of them, and it helps us to get to a better solution.
So a hybrid of Deanna Troi from Star Trek and Professor Xavier on all benevolent. There you go.
That's a very nice way of putting it. I think it's how has it been for you and your lab and your environment over there to go through this year, and how have you supported the people in your lab through these tough times, in your opinion?
Well, I've tried. Whether I've succeeded, I guess you'd have to ask them. I think it's difficult, especially when, of course, you're talking to people, well, at least initially remotely. It's harder to connect with people remotely than in person, although I think the improvement of the video conferencing has helped. I think you just have to try to keep people positive and motivated, and trying to get them to focus on what they're achieving rather than the timeline that they're achieving. I think there's a lot of stress with the timeline. Kind of goes back also to a time when I was younger, when I was a postdoc and a grad student. Every week I'd open Nature and I'd see all these papers, and I would just feel stressed because every week I'm seeing something new, and I can't work that fast. I need years, and these people are coming out with stuff every week. But it's important to realize that paper you're seeing each week may have taken several years to develop. So it's not that you're going slower than everybody else. It's just that science takes time. And we have to try to disconnect ourselves a little bit from the time and just focus on each week, have we done something valuable and important and productive? And as long as we feel at the end of each week that we've been productive, we should be happy with what we're achieving.
I think that's a great way to end the podcast, on that note of that very valuable piece of advice.
Stay positive, stay happy.
That's just, we can always hear that. That's always important to bring up, to stay positive. And I think for researchers it can be hard sometimes to do that. Yeah.
And you have to also try to have some fun, So this weekend we're having our Oktoberfest party. I've got 4 German beers on tap. So hopefully that whets everybody's appetite to have fun this weekend, and then be energized when we get to the next work week.
Perfect.
Brilliant. Okay. Thank you again so much for talking to us today. It was a great conversation, and enjoy your Oktoberfest celebration then.
Yes, definitely. And thanks for having me. It's been a pleasure talking to the two of you, to the audience, and have fun, stay safe, and be happy.
Thank you very much.
Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.